So I'd like to start by thanking Eddie, David, and all of JGI for inviting somebody at both the geographic and intellectual fringe. So we'll see if I can keep you occupied over the next half hour. What I'm going to talk a little bit about, this is going to be a very different talk because you're not going to see any models. So you can sort of intellectually lay back for a moment, but perhaps let your imagination go, because what I'd like you to think about is the genomics of resilience, and particularly climate resilience, or resilience to any form of human perturbation. And I think Panama provides a tremendous opportunity for people that are interested in environmental change and in the genotypes of the organisms that occupy those environments. But I want to start out with this slide of the world and point out, sorry, there's a little bit of problem with this is the only slide I think that has a problem, but point out that, of course, what many of you know is that all of the great universities in the world, most of them, are in the North Temperate Zone. So if you look at the top 300 universities in the world, 94% of them are in the temperate zone. But 80% of the estimated plant and animal biodiversity is in the tropics. Moreover, while we have this map of the world, I'd just like to remind you, I think when we think about climate change, and we think about human perturbation in general, that change is most evident at the poles, and the evidence decreases as you move towards the equator. But although subtle, I'll make the argument that in fact climate change will have profound, and is having profound implications in the world's tropics. Also, we have the map of the world in front of you. It's just a reminder that, at least in my view, when you think about landscape transformation around the world, we've transformed the North Temperate Zone. We've transformed it. It's what it is. We're not going to see a lot of change in the next 100 years in the North Temperate Zone. Climate will shift. We'll see growing seasons expand a bit. Agriculture will change. But it will pale in comparison to the transformation that's going on in the tropics right now. So still the predominant signal, if you look at landscapes from space, in the tropics is one of tropical forest loss, deforestation, contributing about 16% of carbon emissions to the atmosphere. But the second signal you see from space is one of tropical forest regeneration. And this has been largely overlooked. And I would submit, as we go from 7 to 10 billion people, how we use these landscapes in the tropics is going to be critically important to human well-being, human welfare. And we're not thinking about it sufficiently. To give you an example that draws from the earlier talks, Chris's magnificent plenary last night, Wayne's presentation they just provided, you're all thinking a little bit because of those talks about nitrogen. Well, nitrogen in the temperate world, in temperate agriculture, is in short supply, and it's obviously an essential nutrient. But when you move into the tropics, soil nutrients and their importance changes dramatically. Nitrogen is relatively unimportant. Phosphorus is critically important. And I'm going to leave that as a tease because I'll come back to that at the very end of the talk. But it is a fundamental difference between the temperate zone and the tropics. And in Wayne's talk, I wanted to ask him, but I wasn't quick enough. In the very nice slide he showed of the grass growing back, was phosphate added to that experiment? Because in Western Australia, the soils are famous for being deficient in phosphorus. And how plants, particularly the Proteaceae, have dealt with that is in itself a really interesting biochemical story. So one of the things I want to do is just give you a little bit of background about Panama and me. So this is the Isthmus of Panama. I've lived there for about the last 23 years or so. And I was really initially drawn to Panama. I'm a geneticist by training. Went down there to develop a lab. And I was drawn to Panama by two spectacular experiments that I'm just going to touch on now because I think it's of general interest and everybody should know it. But it's not really the purpose of today's talk. But that narrow isthmus connected South to North America roughly 3.5 million years ago. There may have been a ephemeral connection about at the end of the Miocene, so 5 to 6 million years ago. But really it joined South to North America 3 million years ago. And this created this tremendous biological exchange. And for somebody like myself who likes to understand something about the assembly of communities from both neutral or niche-based models, this was an incredible opportunity to look at this from an experimental perspective and understand something about the assembly of communities. So a wonderful natural experiment. The second natural experiment that was created by the rise of the isthmus, you can see quite clearly here, was the separation of what was a continuous tropical sea into the modern Caribbean and Eastern Pacific. And again, as somebody's interested in community assembly, diversification, the origin of new species, this was also a tremendous opportunity. And early in my career, I did a lot with molecular clocks. And of course, having something like this, which is a typical Meyerian dumbbell model of speciation, where you had very large populations that were split by the rise of the isthmus, was a wonderful opportunity. But Panama, as I'll go through in this presentation, also because of the variation in the environments, the impacts of things like El Nino, La Nina, is a remarkable place to think about how organisms adapt or acclimate genotypically to environmental change. And Steve Palumbi, as I've listened to the talks this morning, it seems that people on the other side of the bay are not represented here. This seems to be a Berkeley crowd. But Steve is an old friend and on the faculty at Stanford. All right, we got a couple, that's good. And he was in Panama in October. And one of the things that we do, I think in a really cool way in Panama, is really big experiments. And Steve and I were talking, and Steve was like, we've got to do some really big common garden experiments, because if you think about it, at least some of the solutions to climate change, or again, any human perturbation to the environment, likely already resides in the genomes of organisms. And in Steve's own words, our job is to find those and put them to use. And so when you've got a setting like Panama with tremendous environmental variation, it provides a tremendous opportunity to actually investigate how organisms are responding to the environment. And that's really what I'm just going to introduce you to today. So one of the things about Panama that's fascinating from an environmental perspective are these two oceans, but in point of fact, they differ dramatically even within the Caribbean. When you're in the Bocas del Toro region, which is western Panama towards Costa Rica, as you can see, you've got tremendously high freshwater input, and therefore the marine setting is lots of organics. Whereas in San Blas towards Colombia, you've got a much more oligotrophic setting, much more similar to what you typically think about in coral reef environments. You cross over the isthmus to the Eastern Pacific, and in the Bay of Panama, that big block to your right towards the east, seasonal upwelling, very strong seasonal upwelling, in fact. Very high productivity, whales, huge fish, whale sharks, everything else. And then you go to the other side of the Azuero Peninsula, that block that steps down there, and you've got basically a non-upwelling zone. And that'll become important in a moment. But first, let me introduce you to something many of you know about, and actually that JGI has contributed to, which is this thing we call the coral holobiont. So of course, Panama's in the tropics. Both in the Caribbean and in the Eastern Pacific, we have coral reefs. Coral reefs, like the next subject I'll discuss, tropical forests, are structural entities. And they're incredibly important structural entities. And right now, they're at risk. So from a genome perspective, there's tremendous opportunity here to think about how these coral holobionts are responding to climate change, to human perturbations. And many of you have heard, obviously, about ocean acidification and the impact that it's likely to have on an organism like this that builds its mass through calcium deposition. But there's also some really cool biology to come out of just thinking about the genome. So obviously, many of you are aware that the coral holobiont is an animal, the coral, and a plant, this microalgae here called zooxanthellae symbibiodiodont. And how those two organisms converse in the cell is, of course, fantastically interesting, particularly because at times, and you'll see a picture of this in a moment, the corals throw out the zooxanthellae or the zooxanthellae choose to leave. We don't even know that basic information about the interaction. Recently, really within the last decade or so, this coral holobiont has obviously become more complex because in addition to the dinoflagellate and the coral animal, we now have bacteria and viruses to think about and how they interact. There's been some nice study, insufficient study so far, to suggest that you do get species-specific bacterial assemblages with some of these corals. And so again, thinking from a genome perspective about how this holobiont is going to respond to change is really a tremendously difficult problem. One of the reasons that I'm not showing any models here is because when you get into the tropics and you get into these systems, it's not so easy to draw those theoretical models, although I've enjoyed looking at all of yours. So again, you go back about 15 years, not that long ago, and really we had a very fixed typological view of corals. We had coral species. We could say something about their distribution, something about their response to environmental variables. And we obviously knew that they had these dinoflagellate endosymbionts with them. But at that time, we thought that there was a single species, globally distributed, of endosymbiont. And work by Rob Rowan, Nancy Knowlton, and others demonstrated that that wasn't the case. And this is from a paper of theirs in Nature, more than a decade ago now. But it's still, I think, very successful in demonstrating why this is important. So again, this only shows two species of Montastria, which is one of the large reef-building corals in the Caribbean. There's actually a third species. But when you start to map the dinoflagellate, the zooxanthellae distribution on top of the corals, you get something very interesting here. And what you see, and what's shown in the histogram, is the relative abundance of 3 different dinoflagellate genotypes. Okay? Again, not known 20 years ago, now known. And what you see is the distribution of those endosymbionts changes with depth and with species. It also turns out that it changes with any sort of environmental challenge. So you can actually observe in these species that when there's a warming event or something like that, the distribution of those endosymbionts will change. Again, we know nothing about the bacterial dynamics that are superimposed on top of that. But it's incredibly interesting. And again, whether you're thinking about this from a straightforward biology of symbiosis, biology of mutualism perspective, or thinking about it in terms of the response of these holobionts to environmental perturbation, it's a fascinating area, I think, for continued discovery. In one, as I've listened to most of your talks, that recognizes that if we're going to make any headway on this, we've got to start doing this in a much higher throughput way than we've done in the past. I mean, these studies are largely carried out by people like myself, ecologists and evolutionary biologists, not really people that were trained in molecular biology, although I was to a certain extent. And we tend to think small. I mean, I think, Chris laughed about, his synthetic construct costing $36,000, And ecologists and evolutionary biologists love to get those types of funds in. But if we're going to do what Steve Palumbi and I want to do, we're going to have to coordinate with groups like JGI and really think about how we do this in a massive way. So here's a picture of a bleached coral. In the Caribbean alone, in the last 30 years, we've lost 80% of coral cover, probably mostly related to increasing sea surface temperatures during the warmest times of year. And what you're seeing here is a coral. The animal's still there. It may or may not still be alive. But it certainly has no longer got its microalgal endosymbiont. associated with it. And again, figuring that out, why that happens, exactly how it happens, what is the conversation between those two organisms within the coral, something we know very, very little about but need to learn more about. And this just shows, this is actually from the Eastern Pacific, this is work of Peter Glynn's, but it gives you a sense of the diversity of response of corals to environmental change. And so what you see graphed on the vertical axis is sea surface temperatures, number of reef provinces where you have severe bleaching. And then across the horizontal axis, you've got time. Another sort of hallmark of studies done at the Tropical Research Institute are long time series. We're really good at big experiments, and we're really good at following them through long periods of time. But what I wanted to demonstrate with this slide is just variation, because again, partly what I'm interested in is how do you use that natural environmental variation to point to genotypes or genomes that might be successful in dealing with perturbation. But this becomes clear on the next slide. And so here you've just got, on the horizontal, you've got 2 years mapped. And now on the vertical, you've got sea surface temperature. And what you're looking at is the 97, 98 El Nino in orange and the 82, 83 El Nino in yellow. And you're looking at the Gulf of Chiriquí, Panama, and Gulf of Panama. So Gulf of Panama, from that earlier slide I showed you, is the area of extraordinary upwelling, and the Gulf of Chiriquí towards Costa Rica is an area with much less dramatic upwelling, or many years no upwelling at all. And what you see in the bars at the very top is whether or not there was bleaching or not. And you can just, by thinking about the differences in sea surface temperature in the two graphs, the fact that the two graphs represent two different geographies, you begin to get a sense of the variation in how these coral holobionts respond to those environmental perturbations. It's hard to make sense of it just looking at it like that. There's other things going on. It's not as simple as just sea surface temperature reaches 30 degrees Celsius, or it's upwelling versus non-upwelling. There's more going on. And that's the challenge, I think, for all of us that are interested in how genomes respond. All right, so now I'd like to change gears. Again, one of the things I'd like to have you all leave with is this idea that one of the things I'm interested in is structure. So the structure of a coral reef. I mean, these are incredibly important for human welfare. They provide the structure for fish, et cetera, protein for humans. Other big structure in the tropics is, of course, the forest, the trees. And so I'd like to now spend a little bit of time discussing that. And again, Panama, from a geographic perspective, an environmental perspective, is very, very interesting. So you can see, and I'm not going to read it for you, but you can see that rainfall varies dramatically across the isthmus. And you can also see that elevation changes dramatically across the isthmus. And I'll have a refined map that gets a little bit more interesting in a moment. And again, just to draw reference to this El Nino-La Nina event, you see in the light blue, you've got El Nino, and the yellow, you've got the La Nina event. And what you've got graphed on the vertical axis is the number of species fruiting. So this is the work of Joe Wright. Again, you see that very long time series of observation. And what you notice is that I should mention that El Ninos in Panama are marked by less cloud cover, less rain, although it's actually the distribution that changes. So it's a longer dry season. Our dry season starts in December, typically ends by April, but in a strong El Nino, it will go to June. And with that increased solar insolation, we have increased fruiting. Then, and this is a very complex but interesting outcome of these long-term observations, in La Nina, and particularly extended La Ninas, you get much, much lower fruit set on these trees. And oftentimes in the forests, in the jungles of Barrow, Colorado Island, where we do a lot of our work, you actually get famine. you get tremendous mortality of forest animals, monkeys, sloths, ocelot, et cetera. But the main point of this slide here is, again, just to remind you that in this small area, because we have these La Nina, El Nino events, we're seeing a lot of what we anticipate will be the future of climate change. So again, back to rain, we think in some areas rain will increase in frequency, increase in intensity. Other areas there'll be an increase in intensity but no increase in frequency. Other places, less intensity, greater frequency, every sort of mix you can imagine. And in Panama, you get that. And I'll show you in another map that'll become even more clear. And so a related question is sort of what drives tropical diversity. This is something we've been looking at for a long time. And to sort of make it symmetrical with the coral reefs, again, when I first got to Panama, a tree was a tree was a tree. It was only relatively recently that people began recognizing that, no, these trees all have endophytic fungi. And what you're seeing here, for those of you who enjoy chocolate but don't know what it looks like in the forest, that's a cacao pod. Cacao used to be hugely produced in the New World. The genus is a New World genus. Most of your chocolate now comes from Africa and Southeast Asia, but the genus and the species is native to the New World, and many people would say the best chocolate comes from the New World. But it ultimately fell to tremendous fungal infections. But if you just look at, now I'm not looking at the cacao pod, which is what's impacted in agriculture, but just the leaf, and this is work of Alan Harries. And this is the old way of doing things, right? Just going out and culturing on a plate. But that's from one cacao leaf, and that's some of the fungi you see there. But every tree in the forest has endophytic fungi associated with the leaves. And again, understanding something about that interaction between the endophytes in the trees is something that's an extraordinary opportunity for students of mutualism, but also in thinking about how plants, in this case, respond to environmental change. So now we're just looking at central Panama, and that's the route of the Panama Canal there. And the biggest island in the lake, right there, is Barrow, Colorado Island, which is where the Smithsonian started 100 years ago. And I should digress for one moment to tell you a little bit about us at the Smithsonian. So we've been in Panama. We're part of the National Museum of the United States, but we've been in Panama since 1910, when the U.S. followed France in constructing the Panama Canal. We're a research institute of about 400 people, 45 PhDs. Most of us are ecologists or evolutionary biologists. But look at this. We're talking about an 80-kilometer distance between the Caribbean and the Eastern Pacific here. And look at some of these changes. So you've got a twofold gradient in rainfall across that 80 kilometers. You've got phosphorus that changes by about 200-fold. This is basically the extremes of soil phosphorus in lowland tropical forests around the world. And we have it in 80 kilometers. Huge variation in organic carbon, pH, and this owes to the very complex geology of this area that's led to these soils, mainly clay soils, but clay soils with tremendous variation in nutrients. And so actually this week, or last week, online, it was out a couple of weeks ago, we've just published some work in PNAS. And this is really an extraordinary graph, and I think I'm on time, but I am almost, this is the next to last graph. But it's worth looking at. And basically what we did, this is largely the work of Rick Condit. In that previous map, 30 years ago, we created a 50-hectare plot on BCI. And we followed the species dynamics for the last 30 years. So we go back every 5 years and we completely recensus. We know the botany of that area very well. And so Rick Condit, who's been involved with that for many, many years, decided that he would put in 100 one-hectare plots across the Isthmus of Panama. And those one-hectare plots vary in vary in species diversity from 65 species to about 155 species, something like that, which is remarkable. I mean, across the whole area, there's 550 species, many more than you've got in the entire temperate United States. But what's cool about that is by doing that, by going out and doing very careful soil surveys, they were able to actually look at species-specific responses to resources. It's never been done before. Nobody's ever been able to sort of ask a question of species in the tropics and say, what controls their distribution? So they looked at 8 variables, and to keep this short, I'm only going to talk about the 2 principal ones, and they're remarkable. One was not a big surprise. It's moisture. It's really the amount of moisture that falls during the driest time of year. All right? That explained distribution for more than 50% of the 550 species that were looked at in a statistically significant way. And those are the blue dots, okay? So you've got some species that like low moisture and some species that like high moisture. And then in the open red circles, you have the big surprise. And I sort of teased you with this early on. That's phosphorus. So more than 50% of the species showed a very strong response to phosphorus. And as you can see from the distribution of points around the graph, you have every possible combination. So you've got some things that like a lot of phosphorus and some things that that need very little phosphorus. And I'm just going to make this biologically relevant, this is a species here that's a Canopy Emergence. So the canopy in Panama is about 40 meters tall. Canopy Emergent would be a 50 to 60 meter tree. That's a big tree. It's deciduous, and so at this time of year in Panama, you look on the hillside out in front of my house, and you see that particular species everywhere. But it's weird. I mean, you expect it to be found in places that are dry. It's deciduous. That's one response to dryness, is to shed your leaves. But that wasn't sufficient to explain it. But this graph does explain it. You find it actually on very phosphorus-rich soils. And that seems to be the key. And then when you start to think about the significance of that, it starts to make sense. Well, these are trees that are turning their leaves over all the time. They might need phosphorus. I already indicated earlier that nitrogen tends not to be limited in tropical soils. It's phosphorus. And so that's really cool because for the first time, we have species-specific responses to these types of variables. That means you can go out now and start to do experiments on physiology, genomes, et cetera, to understand and begin to build real models of how we predict forest composition, and not only forest composition, but forest function will change, again, with environmental perturbation. And that would be a first in a place like the tropics. And it's a wonderful experimental opportunity for somebody a lot younger than me, and actually a lot younger than lots of people in this room. What's also cool about this, many of you might have seen, there was an article in Nature in November by Jeremy Grantham. Jeremy Grantham runs a major asset management firm. I think he's got about $100 billion under management. And he wrote an article in Nature saying that most scientists were missing the boat. We were focused on increasing levels of carbon dioxide in the atmosphere. Water was a concern. And he said what we really need to be worried about as we measure with increasing detail increases in carbon dioxide, loss of water, et cetera, was phosphorus. And he said that because there's limited phosphate being mined in the world. And I wrote him a letter because he's a donor of mine and a friend. And I pointed out to him that in the August issue of Nature, there was a description of pistol, which is phosphorus tolerance starvation gene. And that paper just very quickly pointed out that if you look at traditional rice varieties, you find this gene, and lo and behold, it enhances root growth, and with that phosphorus uptake, and that rice can grow on very phosphorus deficient soils. But if you look at the rice reference genome, that gene is absent. And so again, it's a call to recognize that a lot of the things that we've done, particularly in agriculture, and if you're an evolutionary biologist like I am, it makes sense. We've lost genes of significance as we think about how we're going to feed a future world. And then my last slide and point is to remind you that the Panama Canal goes through this area. Panama Canal represents 4.5% of global commerce. And in fact, the canal, whether managed by the US or Panama, has always just focused on the engineering aspect of it. But it sits in a 300,000 hectare context, environmental context. And I think it's a remarkable opportunity to bring commerce of the world to recognize the importance of the type of work that we all do to understand genotype by environment interaction and how it's expressed as phenotype. And so with that, thank you very much.